Thank you guys for tuning in. My name is Nathan Kim, and I'm a rising junior at Stanford. And this summer, I was able to conduct research in the Dixon lab on ferroptosis. So the title of my research is called EMP2 Inhibition Sensitizes to Cell Death. So I'm going to quickly go through my poster here just to summarize the research that I was able to do this summer. Starting with background, what is ferroptosis? Ferroptosis is an oxidative form of non-apoptotic cell death. The process is dependent on iron and is driven by the accumulation of lethal lipid peroxidation. And then RSL3, which is a drug that I used in my experiment, is commonly known to induce ferroptosis. Quickly going through the rest of the background here, palbociclib, or otherwise known as palbo, is known to promote ferroptosis. In this lovely chart that was actually conducted by my mentor, Jason, in the lab, you can see that as the dose of RSL3 increases, the lethal fraction increases as well. Moving to the top right, we can see here that gene expression of EMP2 is impacted significantly by palbo, which is why in my experiment, we decided to focus on the gene EMP2. And then finally, you can see here is our working model that we were basing this experiment off of. And the whole goal of the experiment, as you can see by the question mark, was to determine whether or not EMP2 induced or inhibited ferroptosis. And the experimental questions in my experiment was: 1, does inhibiting EMP2 sensitize to cell death? 2, is the death observed ferroptosis or another form of cell death? And 3, is ferroptosis more sensitized by EMP2 inhibition or by palbociclib? Now, quickly going through my experimental setup, my experimental setup had 3 main steps. The first of which was siRNA transfection of EMP2. After transfecting the cells, and you can see on the left here all the controls, and on the right is the actual layout of my plates for my experiment. After that, after waiting 48 hours for the palbociclib to get incorporated into the cell, I then drugged the cells with RSL3 and also using fer1, which is a compound that is known to inhibit ferroptosis. After doing this and scanning every 24 hours up to 48 hours, so 3 total times, we did the third step, which is a qPCR to confirm the results. And what the qPCR told us was whether or not the siRNA had successfully gotten into the cells. It was more of a check the results sort of experiment, just to confirm that everything had gone smoothly. Moving up to the results here, you can see that figure 1 is the results from the siRNA transfection experiment. And we see 2 main points here. The first of which is it looks like the palbo did work. As you can see that the overall lethal fraction in the palbo, the 2 rightmost columns, are much greater than those in the DMSO columns. And the second more interesting result that we found was if you look in the DMSO plus fer1 column, you can see that we would initially expect that all 3, both the black, the blue, and the green bars would all be fairly low, as we would expect the cells to be dying via ferroptosis, and therefore, since fer1 is the compound incorporated here, we would expect that all 3 of these would not be dying, since fer1 inhibits ferroptosis from occurring. However, you can see that while the black and the green bars are extremely low, meaning that those cells aren't dying, the black, the blue bar, and rather the siEMP2 13.1 cells are still dying, which tells us that for whatever reason, the siEMP2 13.1 cells are dying by some other means other than ferroptosis. So whether this be, apoptosis, or whether this be some other form of cell death, that's one very interesting result that we found. Moving to figure 2, this is the qPCR that we did, and these results are very positive. It tells us that the siRNA successfully got into the cell. As you can see, we would expect that the siEMP2s would be much lower and have a much lower fold change than the negative control, which is what we're seeing in these results. And then finally, in figure 3, you can see that these are 3 different images I took of the cells from figure 1. And just to confirm our results, you can look at the middle image. You see that those cells look less healthy than the cells on the left in the negative control, and the cells on the right in the 13.3 cells. And finally, just concluding some of my original thoughts. Looking at this new working model, we have identified that EMP2 inhibits ferroptosis from occurring. And just to reiterate, one of the very interesting points we saw was that 13.1 was dying by some other means other than ferroptosis. So that is definitely what we would want to spend our next steps investigating further. Thank you.